The cell, biology, and biomedicine. The way I think about this issue is the following. That in biology, what one is fundamentally interested in is the cell because it's the smallest thing that's alive. You're interested in life in biology because biology is basically the study of life. That's terrific. If you're interested in medicine, you'd like to know how cells connect to things like hearts and brains and livers. But a biologist is fundamentally interested in this. Now, this object is made up of proteins and lipids and carbohydrates and whatever, molecules. And the amazing thing about life, and one of the great questions in modern natural science, is how do you go from molecules and reactions, which are molecules and reactions, to the cell, which is alive. Something happens in between here. You know, things become alive. And there's a very, very interesting series of subjects which stem around the fact that we have two words. We have alive and dead, or alive and not alive. But are those the only possibilities? I mean, how about something that's not quite alive, or a little bit alive, or alive but not alive? I mean, the fact that we have two words doesn't mean that that's the way it is. And there's probably a continuum between alive and dead. So, for example, I take, better to take me. Take me. Soon I die. And I don't die all at once. It isn't the bulb goes out, brain begins to cease to function, but my hair, if I had hair, would continue to grow for days afterwards. So parts of me are alive and parts of me are not alive. But even the parts that are alive are probably chemical reactions that are just sort of going on for a while. And this question comes up in a very, very interesting way when you ask, how did it get started? You know, what's the origin of life? Because life, as far as we understand now, came out of a pea soup of reactions in some ghastly primordial planet. And out of that came this, and Beethoven somehow. We don't know how any of that happened. Anyway, the point of interest is that if you want to look at this, which is a couple of microns across, you need probes that can look at small parts of it. So if you look at the cell in greater detail, here's a cutaway, a schematic, and there's the nucleus down in there, and the Golgi and mitochondria and various fibers and stuff like that. That's an electron microscope picture of the same kind of thing. And you need to be able to look at pieces like this. You need probes that are nanometer scale probes. So I want to just show you a couple of my favorite animals. This little fellow here is, back here he has a rotary motor that's churning around furiously, and then this exotic motion here consists of building structures internally that are fibers and then sort of pulling himself along. It really is the self-levitating elephant. And so he, she, it, they, whatever the right answer is, illustrates two kinds of motors. One is linear motor, which is the motor that makes your muscle run, and then a rotary motor, which is big for bacteria. And here's the structure of this, and it's truly astonishing. And it's related to another structure, which is, for those of you who are in the biology business, one of the most amazing things that I know. This is the ATPase that makes ATP in the cell. There's the lipid membrane here in schematic way. And here's a structure that sits in the membrane that rotates. And here's a shaft with various things attached here. Here's a kind of catch that catches as it goes around. And then as protons from the outside stream down into this structure, make it rotate and flip out here, this entire business is driven in a circular fashion that causes changes in the conformation of these proteins down there, and that takes phosphate and ADP and makes ATP. Unbelievable structure. Here's another one of my favorite structures, which is mitosis. And this is a picture I can watch indefinitely. There are all the chromosomes. And here they all somehow get organized along structures that you can't see, the actin filaments, and lined up the right way and then yanked into two separate sets with the right coding of each, so that ideally you end up with the same set of chromosomes in mother cell and daughter cell. And then after that's happened, chunk, you form a membrane down the middle, and this all dissolves to start making proteins into the nucleus. Now, all of that stuff is going on at the scale of a few nanometers. Those are tens of nanometers across. They are hundreds of nanometers. So you need those kinds of small scales. And that's one of the things that's coming out of the current enthusiasm for nano, tools that enable you to look at structures on that size. Here's a biomedical application. These are particles of magnetite. These particular ones are about 20 nanometers across. They're very biocompatible. You inject them into the bloodstream. They don't escape from the vasculature, but they change the magnetic properties of the water in the blood. And in magnetic resonance imaging, you can light up the vasculature this way. So this is a picture for this patient of the circulating blood in the brain, and you can see that if you were looking for an aneurysm or a tumor or something like that, you'd love to have that kind of information. All right, now, this is an interjection, the nature of revolutions. We say that nano is revolutionary. What is revolutionary? That turns out to be a pretty interesting question, and one which people who are interested in the philosophy of science spend a lot of time futzing with. One way of thinking about this is to ask about some things that we would agree are revolutionary. The wheel is revolutionary, fire, bronze, whatever. And then at present, in terms of technologies, cell phones and radar and nuclear weapons, those have all been revolutionary in one or another fashion. The World Wide Web, and in the future, intelligent machines and highly sentient animals. Would you like to have a pet that is significantly brighter than you are? Would a pet that is significantly brighter than you are like to have you? Which is another more interesting question. But these are the kinds of things that technology raises. So where does nano fit in that? Well, here is an example of something that happened. This object replaced that object. And this object and that, the chip plus the fiber replaced, for better or worse, the letter. Although my wife points out to me that the fact that all email messages are essentially public, in her opinion, is going to bring back the love letter, because it's the only thing that you can do. A piece of paper is the only thing that you can write on that actually has some privacy associated with it. And here's the cell made up of objects of that sort, which are molecules. So which is it going to be? Is this going to be a really big deal comparable to that, or is it a little deal? We don't know the answer to that yet. There are, in the theory of revolutions, two kinds of revolutions. There's a revolution which is triggered by the invention of new tools. So the invention of the atomic force microscope, the scanning probe microscope, was a revolutionary tool because it enabled us for the first time to see individual atoms and molecules. Done. That's happened. So we have a revolutionary tool here. Inexplicable observations. There is a theory of revolution that says that science accepts revolutions only when it has to. And so the question is, are we in a situation with nano where we're forced to? And there are a number of parts of this, understanding how the cell works and understanding quantum phenomena and things of that kind. Our back's up against the wall right now. We can't really understand what's going on. So I'm actually optimistic that for all of our torpor, this area is going to force us to rethink some things. All right, now back to some of the other subjects. Information, absolutely a big deal. When you think about information, it's good to think about it in a very broad sense. All of these are different forms of information. This is binary. That's, I don't recognize the tune, but anyway, that's musical notation. Here is information coded in the 4 bases of DNA, and then there's information just in the position of these atoms and lots of information in that. And in fact, the whole subject of information, for those of you who are interested in teaching students, it's very good to raise with them what the word means. Everybody knows what information is until you stop and actually ask yourself what information is. And since it basically is running the world now, it's not a bad thing to bring up as, what is information? And there's very elaborate and interesting theories constructed originally by a man named Shannon to understand that. Now, information is manipulated in the modern world in 2 ways. It's bits of electrons or pulses of electrons flowing around in semiconductor circuits, And then it's packets of light being transmitted through optical fibers from one place to another. So basically, most transmission now goes on optically in terms of fibers, and most processing goes on in terms of electrons in silicon. And the sizes that are permitted here are fixed by the sizes of the photons that are being transmitted. Here, the idea is that you can make things smaller and smaller. The basic way in which one makes silicon chips, I know is familiar to all of you. Photolithography, basically photography, that enables you to draw lines of different kinds in photoresist or on circuits, is the technology that's the underlying basis for microelectronics. And it is a technology that is right up there with bronze and the wheel. In fact, I would argue that it may be the most sophisticated technology the human race has ever invented. It's just phenomenal. And the entire of the information world is based on how that works. And it makes very small structures. And the underlying idea is that you do, as I say, photography with ultraviolet light. You shine light through a pattern. The pattern is then run through a lens, and the lens condenses that pattern into a small size, and various kinds of chemistry happen here that end up making a silicon chip. What's been so interesting about this is the supposition has been that the size that could be made this way was limited by diffraction. And it is. But you can tinker with diffraction. And so you use, instead of shining the light through air, you shine it through water. That gives you a factor of 1.5 in size. You use shorter wavelengths of light. Things get smaller. And one can now make structures commercially in which the lines are 40 nanometers at that point. And there's a roadmap that will get us, we think, to 20 nanometers. So this is a production transistor. The details are not important. What's important is simply that that feature size there is about 40 nanometers. So we'll go to half that pretty clearly in the next period of time. And then at that point, nobody knows what happens. I don't know whether we're going to go beyond it. But you know, physics tells you that you can't have a processor that's smaller than an atom, so we know there's an end at the end of this road. And how we get there is a little bit unclear, and how much money people are willing to spend to make things smaller is not quite clear. But when you think about this area, you should really think about that curve, because it's an interesting curve. This is the cost of a fabrication line. You know, we think about lithography in terms of making things smaller and smaller. Why do you want to make them smaller and smaller? Well, actually, you don't care whether they're smaller and smaller. What you care about is they're cheaper and cheaper. And so the whole idea is to make your device there cheap. You want it, well, it's heavy, but after all, your fingers are not going to get smaller and smaller. So at a certain point, that footprint is fixed. And so what you find here is that you can make things smaller. There are real disadvantages to make them smaller because you generate heat when you run a microprocessor. And if we took that thing and tuned it up to the highest speed it could run, right now, it would, running continuously, convert the temperature in the inside of the chip to about 1,000 degrees C and melt the silicon. That turns out to be bad for performance. Right. Expensive, right. Well, this is the real expensive. The factories to make these follow a curve of this sort, and the guess is that in about 2015, one of these factories will cost in the order of $100 billion. And I won't do the calculation for you, but if you want a 20% after-tax return on investment in a world in which the deepest moral value is return on investment, it's hard to do. You've got to sell a lot of chips. And the number, which is a couple of years old now, but it still continues to astonish me. The number of transistors made in the United States now per second is, and there's a brief pause while you guess internally how many transistors might be made in the United States per second. But the answer is getting on probably toward about 10 billion per second. And you say, what do you do with 10 billion transistors per second? And I think you make lots of really smart toaster ovens. I don't really have a very good sense for that. But it's a lot of transistors. Transistors are really exceptionally stupid, I should say.